So, welcome. I think we're officially starting now. Is that correct, Catherine? Yes.
Okay,
Okay.
good to go.
Well, I want to welcome you all. My name is Andrew Hipp, and I'm the current president of the American Society of Plant Taxonomists. On behalf of Jennifer Mandel, Catherine Picard, and myself as co-organizers, I'm delighted to welcome you to the first ASPT webinar of 2026. If you are not a member of ASPT already, we are particularly happy to have you and encourage you to consider joining the society. There's a link to the ASPT website in the chat to learn more and become a member. Our webinar series this spring explores the question, what good is taxonomy? Each of the 5 talks will highlight places where taxonomic work affects people well beyond the boundaries of research. ASPT believes that fostering a strong natural history community is more important than ever, and we hope that this webinar series helps to strengthen that community. Each webinar, including this one, will be recorded and posted on our YouTube channel. Take a look at the link in the chat. Please note that our next talk in the series is by Dr. Susan Pell, Executive Director of the U.S. Botanic Garden. Dr. Pell will be speaking on the subject Taxonomy in Public Gardens, Engaging People in Conserving Plants. Her webinar will be in March 6th at this same time. And again, there's another link going into the chat so you can register if you want to register now. If you don't register today, we will be sending out notes about it later, but go ahead and get registered and you'll be set for next month. Okay, finally, to the talk we've been waiting for. We are honored to have Dr. Sandra Knapp as our speaker today. Dr. Knapp is the Director of Research at the Natural History Museum in London and a research botanist who specializes in the taxonomy and evolution of Solanaceae, the nightshade family. She integrates the tools of biodiversity science and genomics to understand and sustain our planet. She's served on numerous editorial boards and has led the Linnaean Society of London, Fauna and Flora USA, the International Association for Plant Taxonomy, and the Botanical Society of Britain and Ireland. Dr. Knapp holds honorary professorships at University College London and Stockholm University. She's been elected to the American Academy of Arts and Sciences, the National Academy of Sciences of Argentina, and Academia Europeana, and she's a Fellow of the Royal Society. She was awarded an OBE in the King's New Year Honors in January 2023, and in 2024, the Engler Medal in Gold by the International Association of Plant Taxonomy for lifetime achievements in botany. Dr. Knapp is the author of 320 peer-reviewed articles and book chapters, numerous popular articles, and several books, including Flower Day, an hourly account of the lives of flowers by University of Chicago Press, which I encourage you to check out. She is passionate about promoting the role of taxonomy and the importance of science for conservation and sustainable development worldwide, and is committed to increasing openness, diversity, and inclusion in science. As such, we felt she was a fantastic first speaker for our series this year. Please join us in welcoming Dr. Sandra Knapp.
Thank you so much, Andrew. That's really kind. And I would encourage anybody who's not a member of the ASPT, please sign up now because this is one of the great organizations in our botanical community because it brings people together in special ways. And I'm truly honored to be the one who was asked to give this the first seminar of this series because in a way, the question that's asked, what good is taxonomy, is what kind of a question is that? Of course, taxonomy matters. So what I want to do today is take you through a little bit of my journey of taxonomy and show you in a few kind of stories why taxonomy is absolutely essential to the rest of biology, but also to answering some of the really important questions that we have that go into sustaining the planet. But first, before I start, I want to thank people. Science is a village, and we can never do science on our own. And these are just some of the people who've helped me along the way and whose work I'll talk about today and whose ideas have fueled many of the ideas that I've had. I don't think any idea I've ever had is original to me. I think they've really come through community collaboration and talking to other people. So I just want to thank all of these people and many more right up front. Now, I want to start out by talking about diversity. Now, diversity is not distributed evenly. We know that plant diversity is not distributed in a uniform way across the planet. This picture from Neil Brummett's article from 2020 shows that most plant diversity is found in the tropics, and we all know that. We also know that that plant species diversity within genera of flowering plants is also not distributed evenly. There are lots and lots and lots of genera which have very few species, and there's very few genera which have very many species. These big genera have always been known as monsters. So genera with more than 500 species, and there are 86 of those, account for 25% of angiosperm diversity. And those genera with more than 1,000 species, of which there are 28, account for almost 14% of angiosperm diversity. So it's actually really important that we understand and study these big genera. But actually, that's a challenge because these big genera have always been thought of as black holes. There's something that you shouldn't step into because you'll be sucked into outer darkness and will never reappear. And so they've been things that taxonomists in general have steered away from because they are something that can't be done in a 3-year, a 4-year PhD, a 6-year PhD, a 3-year grant period. They're scary. They're scary. But never let anybody tell you that you shouldn't do something. So I want to go back and just talk for just a minute about what I think taxonomy is, because we've asked at the start of this webinar series, what good is taxonomy? So let's think a little bit about what taxonomy actually is. And I always think of taxonomy as like a 3-legged stool, and you can't sit on the stool or do anything unless you have all 3 of those legs. And the first of those, which most people think of taxonomy as being, is description, is describing and delimiting species. Some of these species are new, some of them we know already, and we're just changing our delimitation of them. The next one, which people often call systematics, but for me is part of taxonomy, is phylogeny, is looking at the evolutionary relationships of taxa at various different levels. We often think about the species level. And the third leg of this stool, which is equally important with the other 2, is identification, because you need to be able to identify the plant that you have in your hand to really understand it. So taxonomy has often been characterized as a lone man, it's always a man, in the corner of his office, looking through a microscope, describing nature. That's kind of the caricature of a taxonomist. And that's often used in contrast to big physics. This is a picture from the people from the University of Manchester who worked on the Higgs boson, finding the Higgs boson. So physics is often characterized as really big science, has lots of people working on it, whereas taxonomy is just a little man in a corner doing a few things. And this is often seen is because people think it's because physics needs big infrastructure, things like the Tevatron Collider seen here at Fermilab. But actually, taxonomy has big infrastructure too. And in plant taxonomy, the big infrastructure we have are herbaria. They've just been amassed over many, many centuries rather than a few tens of years, things like the Tevatron Collider. But we have something that these big sciences actually don't really have. And we are a big science because the science of taxonomy also has as its laboratory the natural world, which is infinitely interesting and infinitely exciting. So in a way, we're super lucky. We've got big infrastructure, we have big teams of people, we have the whole world in which to work, which is absolutely fantastic. Now I want to switch to kind of the main topic of tonight, which is my adventures with Solanum. And we all know Solanum. Solanum contains things like tomatoes and potatoes and aubergines, eggplants. I've become too English, it's terrible. Eggplants and a host of kind of local crops, which are really important for local nutrition. But Solanaceae itself is much more than just potatoes. There are 103 genera, more or less, about 4,003 to 4,000 species. But what's interesting about Solanaceae is it's very uneven as to species diversity in the genera. One genus in Solanaceae has almost a third to a half of the species diversity of Solanaceae, which is really, really interesting. Solanums and that 1/2 of species diversity, and I've always been interested. I suppose someone asked me once, what's the big question that you've been asking your whole career? And I suppose the big question I've been asking is, why are there so many species of Solanum? And they said, well, have you answered it yet? And I said, well, getting there. And so I'll explain to you a little bit about how we're getting there. So Solanaceae and Solanum grow in all kinds of habitats. They're from the driest deserts of Peru, the high elevation punas, also here seen in Peru, in the transmontane belt of Mexico, the very, very diverse mountains of the Sierra Madre Oriental y Occidental. They also grow, there's solanums, many solanums in the savannas of Africa. There are also many solanums, which I'll come back to a bit later in the talk, in the dry, arid interior of Australia. But the highest species diversity in both Solanum and in the Solanaceae occurs on the eastern slopes of the Andes, where all of that moisture comes across the Amazon and then drops as it hits the Andes. So in these really wet, very humid environments. Solanums also come in lots and lots of different body shapes. I often have arguments with my zoologist colleagues that say, no, no, plants don't have bodies. Of course they have bodies. They have bodies that interact with the environment. And solanum bodies can vary enormously from being tiny little herbs like this Solanum woodellii, which you can see being held in my thumb, has a bud, a flower, and a fruit, and it's all of a centimeter tall, to large trees like Solanum lycocarpum, here seen from Minas Gerais in Brazil, to trees which are quite big and several tens of meters tall. So that's a lot of diversity in body shapes in Solanum. Solanum flowers, however, are very, very to a bough plan, a plan, which is very, very similar. They've got 5 petals, 5 stamens. They're bunched in the middle. They have poricidal anthers, and anybody can spot a Solanum flower from 10 meters away. But actually, because of slight variations in the andrecium, they've been divided up into lots of different genera over time. But when Lynn Bose, my wonderful colleague Lynn Bose, started doing DNA sequencing in the late 1990s, mid 1990s with Dick Olmsted, they found that actually Solanum, all of these segregate genera of Solanum, fell into what was a completely monophyletic Solanum. And of course, we want to recognize monophyletic groups. We want to recognize those groups, all of whom share a common ancestor. And what we found with Solanum is you have a completely monophyletic Solanum with 100% support in these early phylogenies. But it itself, Solanum itself, was divided into 2 main big groups that you could teleport morphologically. One of which is a monophyletic group, is what we call the Leptostomonum clade, and it's what we call the spiny Solanums. Now, they're not technically spines, they're technically prickles, but spiny Solanums has a much better ring to it than prickly Solanums. So we'll just call them spiny Solanums for the time being. And then there's a big paraphyletic group composed of lots of different clades, which are the non-spiny Solanums, things like potatoes and black nightshades, stuff like that. So, and they're all over the world. Now, what's interesting is that these spiny Solanums are completely monophyletic in whatever treatment, whatever phylogeny we've done, they all come out always as monophyletic. But what's interesting about Solanum, and looking this in comparison to Solanaceae, is the red circle is Solanum, is that the lineage diversity within Solanum is also huge within the family Solanaceae. So lineage diversity and species diversity are very big in Solanum, making it really interesting from a taxonomic point of view, if you think of taxonomy in the way that I do. But Solanum's also interesting for another reason. Well, it's interesting just because it's fascinating, but it's also something that makes interesting connections, because many of the species of Solanum are of great economic importance to us, to people, like potatoes and tomatoes. And this took me on a very interesting journey to the genomics community. Now, today we think of phylogenomics, we all do phylogenomics, but back 20 years ago, this wasn't such a big thing. And so when Lynn Bose and Mike Nee and the late David Spooner and I applied for a grant from the National Science Foundation for the Planetary Biodiversity Inventory to basically do a treatment, global treatment of Solanum, we were given the money on the condition that we worked together with the genomics community, who at that time were beginning to sequence the tomato genome. Now, this is the Tomato Genome Consortium founders in 2004 in Wageningen in the Netherlands. And you'll notice that there's not great gender diversity in this group. It's got much, much better. But this was a really interesting journey for me to start out on, thinking about how do you talk to people who don't speak the same language as you? I first went to the Plant and Animal Genome meeting in San Diego in 2004 and sat in a series of talks at which I just wrote down every word I didn't know and filled a page and a half. So it was pretty daunting at the beginning. But there were other conflicts as well, and conflicts that one needed to talk about and talk to people who actually didn't speak the same language. And one of these is that as part of our phylogenetic studies and our thinking about Solanum as a monophyletic group, is that potatoes and tomatoes are each other's closest relatives. Now remember a few slides back, I showed there was a yellow flower that said Lycopersicon on it at the top. Tomatoes had always been segregated as the genus Lycopersicon, which has 17 species, all very lovely, all have yellow flowers, marvelous, marvelous. But they're totally nested within Solanum. And that was a real problem for the people who were working on the Tomato Genome Project, because they wanted Lycopersicon to stay as a generic name. So we explained about how you define monophyletic groups, how you do this, and we wanted to cut the Solanum tree here at the base and have a monophyletic Solanum. And they were saying, well, no, we have to have this genus Lycopersicum. And I said, well, if you did that, you'd have to have all these different generic names. And what do we want to do? So essentially, and this is off the work of Iris Peralta from Argentina, who's now in Mendoza in Argentina. And there were sort of some options for people with Lycopersicon. Is we could have just said, you should follow us, you should do what we want. But we offered them essentially for three main options, in discussion. And we said, you could ignore us completely. That's fine. We're going to continue to call tomatoes Solanums. You could ignore us completely. We could change the names of the potato to Lycopersicon tuberosum and then the eggplant to Androceros melongena. That would be okay. We could do that. Or we could just change the name of the tomato back to Solanum lycopersicum as Linnaeus had originally named it. And there's a long story about how Lycopersicon got to be a different genus, but we won't go into that here. And they thought about this for a while, and then they thought, actually, this is the right thing to do. And one of the really important people in this whole story of tomato genome community and biodiversity people coming together is Danny Zamir and also Zach Lipman here, who works at Cold Spring Harbor now. Danny is in Israel, is that they thought this is a really good thing. And I can remember Danny standing up at a meeting and saying, when we were Lycopersicon, we had 17 relatives, and now we're Solanums, and we have more than 1,000. Isn't that amazing? And I think with that we came together and really thought about it, changed people's minds. And in 2011 and 2012, there were two Nature covers, one of which was the potato genome and one of which was the tomato genome as Solanum lycopersicum. So it just took a while. It takes conversation, and I'll come back to that a bit later. Also working with the plant breeding community, it's been really important to have taxonomy and nomenclature and all of the things that we do as taxonomists as part and parcel of thinking about how to improve crops. And this is work that was done by Xavier Obrio, who when he was doing a postdoc with me in the UK, is we looked at the eggplant clade. So we looked at the group of species which were closely related to the eggplant. And people had always thought that something called Solanum insanum was closely related to Solanum insanum and Solanum incanum. Very unfortunate pair of names, but never mind, that they were closely related to the cultivated eggplant. But in fact, what we found is that Solanum melongena is derived from Solanum insanum. It's a cultivated plant. Solanum insanum is the wild progenitor. But there's a whole slew of other species, most of which are from Africa, which are related to the eggplant and could be really important in breeding. We also thought about, what do you do with crop wild relatives when you have something that's the member of a genus that has more than 1,000 species in it? How do you deal with that? Because what the paradigm was that you have the progenitor and the crop, that's kind of gene pool level 1, and then you have all the other members of the genus, which is gene pool 2. But with Solanum, that's crazy. I mean, that's a crazy thing to do. So with Jaime Prohens, who's from Valencia in Spain and is an eggplant breeder, we thought about what kinds of species could we think about that would contribute to eggplants to make them more climate resistant. And one of the things we thought about was actually, the kinds of plants which we want our crops to be like are weeds. We want them to be resistant to diseases. We want them to grow anywhere. We want them to be tough as nails. We basically want them to be weeds that we control. So what Jaime and his team did is they took a globally highly invasive weed called Solanum elagnifolium, which actually, mind you, is a really lovely plant. Um, and they tissue cultured hybrids with Solanum melangina and Solanum elagnifolium to see if we could create something that combined some of those weedy characteristics. And they had a couple of generations where the fruits didn't really work very well, but finally they got to a point where they have a plant which is bearing fruits and has the genetics of elagnifolium and melangina. And what's really interesting about it is that it combines some of those really good characteristics. Solanum melangina, the eggplant on its own, has a very limited root system, whereas melangina cross elagnifolium actually has a root system which goes out and finds water really well. So doing that has been really interesting about thinking about the phenolics and all of the other things which are downstream in the plant breeding. But it started with taxonomy. It started with knowing about the phylogenetics and knowing about what kinds of things were related to eggplants. I want us to circle back to how we find out about things now. And this is a picture of a lovely Solanum, just because I really like this picture. Solanum has always been a big genus. It was one of Linnaeus's larger genera. Linnaeus had 25 species of Solanum in Species Plantarum. And mind you, at the time, Species Plantarum only had 9,000 species in it, and it was the flora of the world that was known to Linnaeus at the time. So that's pretty good, 25 out of 9,000. But as exploration of the Americas, as botanical exploration and collecting in the Americas ramped up, so did the number of species of Solanum that were known to people. And a lot of this has to do with the explorations that were done by Alexander von Humboldt in the early parts of the 19th century. But a lot of this also has to do with the taxonomic work that was done by Michel Félix Dunal in Montpellier in France. He's really the father of Solanum and Solanaceae taxonomy. He's the one who did it for D. Candole's Prodromus. But in Solanum, we have a real problem, which other genera don't necessarily have. Is we have lots of species. There's 1,253 species now recognized. Well, maybe 254 because there was another one that was described recently. But what we really have as a problem is synonymy. And that circles back to those economically important plants. Is those economically important plants have lots and lots of differences. People would describe them at the level of species instead of at the level of cultivars or whatever they were. So in the cultivated potatoes, which is a paper I did with the late David Spooner. David Spooner was the most wonderful colleague, and some taxonomists at the Vavilov Institute in St. Petersburg in Russia. We did the taxonomy of the cultivated potato, and the cultivated potato has hundreds and hundreds of synonyms. It also has hundreds and hundreds of names which are not validly published, which means it was a nomenclatural morass. And solving all of that is one of those taxonomy tasks, which takes a long time, but goes hidden under the radar. So Solanum has all those kinds of problems, which means we have about 80% synonymy. Across flowering plants, the average is there's one accepted species for every 3 described species, so about 30% synonymy, but Solanum has 80%. So that's been a big challenge for us. And we can often, in some groups, drown in the synonymy, but it needs to be done, and it needs to be done by my generation, so nobody ever has to do it again, to do all that typification and all of that nomenclatural work, which is really important and is often forgotten. But also, working on these big genera means you get to go in the field a lot, which is really, really fun. And it's one of the things that taxonomists, I get asked when I come back from a field trip, is how was your vacation? And I said, it wasn't a vacation, I was working. But actually, in my heart of hearts, it's also a vacation because it's so much fun. And you get to do things like go on crazy roads. You find new things that don't have taxonomic names. We don't discover new species. We find them, we encounter them for ourselves for the first time. But those plants have always been there in their environment. They've been known by the other taxa that grow around them, and probably by the people who live in those areas as well. But we have the privilege to give them scientific names, which is really fun. But all of this, of course, needs to be done in collaboration. Collaboration is the key word to doing good taxonomy. And we'll talk a bit more about that later. And this is a picture of Tina Sarkinen and Paul Gonzalez, two of the most wonderful people with whom I've ever worked. You know, and all that collecting in the field and all that working in those big infrastructures, which is herbaria, also means that we can have data which we can apply to other things. And this is a map. This is the map of the world in solanums, which is an old map because now Australia is much better filled in. But solanums grow everywhere. But if we don't have the data and georeference data to be able to map things, we can't do downstream analyses that I'll talk about a little bit later. So actually sitting in herbaria, databasing specimens, georeferencing stuff is an enormously important task. And we did a paper a few years ago, which I'm not going to talk about, but it showed how important local herbaria were, herbaria in-country herbaria are to actually understanding species diversity in all kinds of groups. So as part of this planetary biodiversity initiative, planetary biodiversity inventory, effort, we did lots of monographs, we did lots of taxonomy, we did lots of field work, we built a website, which is called Solanaceae Source, which I'll talk a little bit about later. The green blobs are things that we've done in the project, and the purple blobs are things that are still being worked on, but actually some of them are very close to publication now. And then down here we have that third leg of my three-legged stool of taxonomy, which is identification. And as part of the project, we also made keys to the clades of Solanum and also keys to the species of Solanum, which are constantly being updated and you can find on our website, Solanaceae Source. And as part of all of this project, we continued to do phylogenetics. So one of the things that often happens is we think of science as being linear, is first you do this part, and then you do this part, and then you do that part. But actually, it works best if you do them all at the same time, and they iteratively help each other get better and better and better through time. So this is a part of the work which is done by Edeline Gagnon, who's now at Guelph in Canada, and Tina Sarkinen, who's at the Royal Botanic Gardens in Edinburgh. And this is our most recent, well, not quite, it's the penultimate phylogeny of Solanum that we have. And this one had 742 species, which is 60% of the diversity. We were really trying to get above half of the species diversity. It was nine genomic regions. And what we found is that the clades, those monophyletic groups that were all in a comb in the phylogeny of Lynn Bose from 2007, they're all still the same clades. Basically, we recover all of the same clades that have been there for the last 20 years. So clades are stable, and we have good groups, but those relationships between them have not been clear, and they keep flipping and flopping around. So the relationship, so that comb-like structure is something that's characteristic of Solanum. And why are those relationships not quite so clear? And it turns out that there are some areas of huge phylogenomic discordance in Solanum. Now, that's often considered to be a problem. But actually, for me, that phylogenomic discordance means here's the biology. Here's the interesting next set of questions. So there's three areas with significant major discordance along the backbone of Solanum. One of which is right at the base, is at the real backbone of Solanum itself, at that backbone. There's at the backbone of the spiny Solanums, there's also wide discordance. And then the other place where there's high discordance is at the backbone of what we now are going to call hemigeocanthos, which we used to call the Eastern Hemisphere spiny clade. Now, one of the things we found out with doing these phylogenetic treatments is that very surprisingly, all of the Solanums that grow outside of the Americas are each other's closest relatives. So all the spiny Solanums outside of the Americas are each other's closest relative. This was completely unexpected. Completely unexpected. We expected these things that were outside of the Americas to be scattered amongst other clades. Not the case, which is really, really interesting. And I'll come a bit more to that in a minute. But I want to come back to discordance a bit. And potatoes, I mean, we know a lot about potatoes. We know a heck of a lot about potatoes because people are really interested in potatoes. We're all interested in potatoes. I had a potato for lunch today, which means, you know, potatoes are great. So one of the places where there was always discordance at a deeper level in the tree of Solanum was at this potato, tomato, tuberosum node. So it's a node of three groups: the potato clade, the tomato clade, and this tuberosum, which is a tiny little clade of three species which occur in Chile and on the Juan Fernandez Islands. And there's always been discordance there. It's always, we're not really sure who's each other's closest relative. And with some colleagues in China who are really interested in potato breeding, their interest is in breeding diploid potatoes. Our interest was in the evolution of these particular groups. So we came together and kind of talked about what we would do in this area. And we did a project together, which was fantastic, which couldn't have been done without the phylogeny, without understanding what the species are, without understanding what were the elements that went into this study. So here's the cladogram, mid-Miocene split between these taxa. And you get a sort of splits tree, which, looks pretty okay. But what they found is absolutely fascinating. What they found is when they went into the genomics, they did whole genomes of all of these species. And when they went into it, they discovered that in the key genes that have to do with tuberization, half of the ancestry comes from an tuberosum lineage ancestry, and the other half comes from a tomato lineage ancestry. So the hypothesis here, and there's a lot of work that needs to be done to test this, and it's a really interesting, so the best science is the science that goes on to ask the next question. And here's the next question: is this really what happened? So the hypothesis is that there was a hybridization, an ancient deep hybridization event between an ancestor of the tuberosum lineage and an ancestor of the tomato lineage, which led to the early potato lineage, at which these two gene families came together, which allowed tuberization. That happened about the mid-Miocene, about the time that the Andes were rising, which means that these new plants with this new power went into open habitats and diversified. And I'll come back to that a little bit later. So with a phylogeny, you can also do all kinds of mapping of characters, which is super fun. And so this is fruit color across the phylogeny of Solanum, and there are 58 gains of red fruits across the Solanum. This is a hugely labile character, which means it may actually be very important in speciation. There are other characters which we didn't map on the phylogeny, but actually look like they're really interesting as well. And this is one that we did together with Zach Lippman's group, doing a pan-genome across Solanum, for which the taxonomy and the phylogeny were essential to get the right sampling. And they looked at locule number, at these kind of cultivated plants where locule number is important. So, when you have a beefsteak tomato, there's lots and lots of different locules. And that's true also of the gilo, or the African eggplant, scarlet eggplant. And what they discovered was that actually the things that govern this locule number increase are different in these different species. And you always assume that something, and this is the kind of Hennigian, looking at things again and again and again, is that one would assume that, lots of locules, yeah, that's governed by the same sort of genes. It actually turns out not to be so, which is super fascinating. But there are some characters that are really, really constant, and they define monophyletic groups. And one of these is prickles, which defines this Leptostomonum group. And there's a single gain of prickles. The most parsimonious explanation is there's a single gain of prickles in Solanum with 14 different independent losses. And this was fascinating also to another set of postdocs and people in Zach's group. And together we worked on looking at species pairs, so sister species, one of which had prickles or spines, and the other of which didn't. And actually within individual species, where species were polymorphic for possession or non-possession of prickles. And what they found was utterly amazing, is they found that the loss of prickles was due to mutations in a gene called lonely guy. I love the way genes are named. They're great. So log, lonely guy, which is a hormone biosynthetic gene. And that's involved in these multiple losses of prickles. But what's really interesting is that it's all kinds of different mutations. And sister species have different mutations. So it's not something so it happens over and over again in all kinds of different ways, which I think, the more you look at something, the more complicated it gets, but then the more questions you get to ask as you go on further. So looking again at this kind of mapping traits onto phylogenies, something that Edeline did based on the paper that Rebecca Hilgenhoff was the lead author on, is to look at geophytes and non-geophytes across Solanum, because we have a lot of we have the potatoes, which have these underground storage organs. And we also have a lot of species of Solanum which have underground rhizomes. Many of these are problematic weeds in many parts of the world. So what she did is she took those characters, those characteristics. Here they are mapped onto the phylogeny. And then what she did is took all of that information from all of those georeference specimens and looked at that against environmental variables to look at the environmental niche envelope of these different types of solanums. And here's the niche envelope of the non-geophytes. It's kind of blobby and is kind of everywhere. The underground storage organs, which is essentially potatoes. Potatoes are the most of the species of Solanum, which have these starch-bearing underground storage organs, or potatoes. Those are in the Andes, and they're in areas which are cold and dry. And the rest of those species which have rhizomes, many of those species which have rhizomes, the rhizomes tend to be in an environmental envelope which is hot and dry. So, so that's really interesting, because that means that these different types of geophytic organs have evolved to do different kinds of things. Now, this was a very crude kind of separation of the different types of underground organs, and it needs refining, but it's a really good look at perhaps how we can expand some of these preliminary studies to ask more deeper, more specific questions about particular things. And work that was done, and this feeds into work that was done by a PhD student, Susie Echeverria Londoño, in 2000, which we published in 2020. And what she looked at was diversification rates in Solanum, because one of the things that one might expect is that where there are more species, there's higher diversification. Actually, the exact opposite turned out to be true. The highest diversification rates are in the areas outside of the Americas. The high species numbers are in the Americas, and the high diversification rates are outside of the Americas. So that is fascinating. And this is the high diversification rate, which actually is completely driven by these outside of the Americas spiny solanums, which occur in Africa and Australia. And if you look at the dated tree of Solanum, and the dating is we're all thinking about dating now, and dating is changing a little bit. But this explosive diversification of Solanums in Africa and Australia is at that same time in the Miocene, when the interior of Australia was drying and habitats in Australia were opening up that were completely different to any habitats that had gone before. And this is true of many, many different groups in the interior of Australia, which is fascinating, and a way of creating kind of a global look across many taxa of what's going on in these rapidly changing environments that are drying, which has real relevance to the world today under climate change. And in a way, Australia is a super fascinating controlled experiment, because there's another clade of solanums here, Archisolanum, which are the kangaroo apples, which are pretty cool. And they occur in these wetter areas in southern Australia and also in New Guinea. And so Australia is in a way a controlled experiment. These are much older. It's an older clade. And so there's sort of different clades have had different experiences in Australia as it's changed over time. So I want you to also think about that green thing. So if we go back one, there's also an area here, which is the diversification rate is higher than background. And that green area is actually the potatoes. So think about what we found with the kind of hybrid, that's ancient hybridization event in the Miocene. The Miocene and the drying and changing of environments has been really important in these different clades and where the highest diversification rates in Solanum are. So that then begs a whole bunch of other questions about what's going on with individual potato lineages within the potatoes, what's going on with particular areas of the Andes. How do we get into this a bit more? So again, another set of questions. So what's important about those two studies and thinking about those and the diversification rates is to really succeed or explosively diversify, you need to be in the right place at the right time, but also have the right traits. So all three of those things need to happen for species diversity to increase. Now I'm just trying to find the thing. Here we go. But, and this is a big but, it's really, really important that studies like that, all of those things using phylogeny, all of those things using environmental variables, all of those things using genomics, actually are underpinned by solid, good, species level taxonomy. That description, but also the phylogeny, but also the identification. Because here's an example. Here's a tree which is whole plastogenomes of potatoes. And this is super interesting, but the red arrows point to things that are all called the same thing. So are those all the same thing? How do you reuse this information if there aren't vouchers actually to figure out? So vouchers go with taxonomy, having something physical to go back to really think about what it is you have. So those collections, that infrastructure we have, those herbaria are crucially important for all of these downstream studies, even though it might seem that they're not. So thinking about over those 20 years of working on Solanum with all kinds of absolutely amazing people, there were a lot of challenges we found at the beginning. So the challenges at the beginning were, are we going to have enough time? There's never enough time. Are we going to get access? Access can be tricky. There's institutional commitment is always a challenge. Institutional commitment to multi-year projects is a real problem. But some of the other challenges that we found at the beginning, things like collective working, credit for effort, common goals, and specimens, are not actually challenges at all. They're actually opportunities. They're opportunities to work with other people. They're opportunities to think about your science in a different way. And the last one of those specimens is, can there ever be too much evidence? I'm not sure. There are a lot of specimens to look at these days, and you want to look at them all, but sometimes you can't. And I think digitization and some of the efforts that people are working on in herbaria around the world are really important to helping with this particular problem. And I think one of our challenges that I think has been the biggest revelation for me is this one of mission creep. Is it mission creep to work with the genomics community? But mission creep actually means you have better reach and that taxonomy gets to more communities and becomes important for more different kinds of study. It also can increase your perspective on the world incredibly. So working on Solanum, although people said, why did you work on something so big and a black hole of taxonomy, has been a way of really expanding. For me, it's been a way of expanding the people I work with, expanding the things I think about, and expanding the way in which I view the world. And that's ever more important now as we reach into the 2030 mission to urgently halt and reverse biodiversity loss. Doing that seems as though it's not connected to taxonomy, but it is. Taxonomy is incredibly important to this. And it's important for us to make the case for this by doing taxonomy and attempting to connect it to other communities, not by telling them they need it, by instead collaborating with them and showing through our actions that it's what we need to do. Because we are at a difficult time. We're at a time where climate change is changing the world around us in ways that we may not be able to predict. We're at a time when anthropogenic change is huge. Wildfires rage. The minute I left Australia in December, there were huge wildfires. It wasn't my fault, I promise. So we're undergoing all kinds of changes. And we as a community, as a community of biologists, community of natural historians, we're quite good at connecting, the global to the local, to the plant, to its DNA. But actually the thing we're not so good at is connecting, having those conversations which allow us to connect the DNA back up to the plant, back up to the community, back up to a global view. So doing that round trip journey is really, really important and requires us to think about collaboration in different kinds of ways. And there's a lot more to collaboration than one might think. We often think of it that we bring different teams together, we have a common goal, we exchange things, we produce stuff together, all very good. But actually, the underwater bit of the collaboration iceberg is equally, if not more, important than the bit that sticks out of the top. We need to treat each other kindly. We need to build those diverse teams. We need codes of conduct so we know how we're behaving with each other. We need inclusive workspaces, places where we welcome people who are different to us. And we need to be explicitly open for contribution in a way that sometimes can feel a little bit scary. But that's the real key to collaboration. And I think one of my favorite quotes, and I want to kind of end with this, is from Alexander von Humboldt, that person who explored the Americas and started this revolution in understanding Solanum species diversity. And he said, the most dangerous worldview is the worldview of those who have not seen the world. And one could take this as meaning that if you travel, you're fine. But actually, what he meant in this particular instance was that it's really important to see the world through other people's eyes. Because in seeing the world through other people's eyes, you can actually have a better, broader, more inclusive worldview. And I think that's one of the keys to collaboration as well. So I want to thank a million people and funding agencies and all the people who've supported this Solanum work over many years. I mean, my colleagues thought I was nuts starting to work on something that had that many species and was that complicated, but it's been a great journey and it's been super fun. And I would encourage all the students who are listening to, if somebody tells you it's not possible, don't believe them. Everything is possible. You just have to think about it in a slightly different way. And I'll end partly with some of my amazing colleagues, but also just give you a glimpse of the new Solanaceae eSource website, which is coming up, which is being built by Tina Sarkinen in collaboration with GBIF, and it will be a GBIF hosted portal. And we're just getting it together. So eventually it'll happen and it'll be on that same URL that you saw a few slides ago. So please go and check it out and tell us how we can make it better, because we want to make this the most useful thing for anybody who's interested in Solanaceae, be it a school child, a government official, or another botanist. So thank you so much for inviting me to give the first talk in this amazing series. And I hope all of you will come to all of the next ones. Friday, for me, afternoons, for you in the East Coast, lunchtimes are a really good time to have these kinds of seminars. And anybody who's not a member of ASPT, join up now. Thank you very much.
Sandy, thank you so much for an outstanding talk. That was truly inspiring, a delight, and really excited. I feel like we all have a ton of work to do now. Um,
Well, I have a ton of work still
yeah,
to do.
we all do. We have about 10 minutes for questions. Is that correct, Sandy? Does that work with your
Yeah,
schedule?
that works for me. Yeah,
Okay,
that works for me.
so I'm gonna let Catherine Picard, who's been
Yay, Catherine.
running behind the
Thank you.
scenes. She made all this happen technically. I'm gonna let her field questions because she's gonna be able to technically handle it more, but I'll be here as well. So, Catherine, I'll let you lay out the ground rules for questions.
Go
Happy to
for
do
it,
so.
Catherine. And
So
thank you for setting all this up. It's amazing.
thank you so much for speaking. If you have a question, don't raise your hand, pop it in the Q&A. We do have a hard limit, so we are going to try and respect that. And so we may not get to all of your questions. Let's start quickly. Would love to hear about the diversity of anthers in Solanum from you.
Oh, wow. Anthers are wacko in Solanum. There's kind of long, thin ones with pores at the top and hairs on the back. There's little short, fat ones. There's ones that look like hearts. They're all poricidal, more or less poricidal at the tips. So all Solanums are buzz pollinated, which is I took a great video of buzz pollination, which one another day. And for another seminar, we'll do buzz pollination. But but the diversity of anthers is really quite interesting. And I always thought it might have to do with the size of bees who pollinate Solanum. So I thought big anthers will have big bees. Well, it turns out not to be. It turns out, as usual with things, I think, it turns out not to be true. Little bees grab onto these big anthers and buzz them. So anther size and shape is quite important in the species level taxonomy of Solanums. And oftentimes in the species level taxonomy, what's also important is the ratio between the anther size and the filament size. So if you think about the fact that there's a kind of resonance that's set up in the anther to squirt the pollen out, then how the filaments and the anther work together is a biophysical thing, which I think is probably warrants more investigation. But yeah, loads of diverse anthers.
Okay. What is the role of phenotypic plasticity and ecotypic variation in the phylogeny of Solanum?
Well, it depends kind of. In the kind of phylogeny overall, we usually don't multiply sample across populations because there's so many species and we want to get as many species as we can into the phylogenies. But many of these species of Solanum are hugely variable phenotypically. I mean, there are some things which people have divided into 10 or 15 species that I've decided to call a single species with a lot of variation. And that's true of the taxonomy of potatoes as well. Potatoes used to have something like 400 species, and in the late David Spooner's monographs, we now recognize 104 species of Solanums. So that variability and phenotypic plasticity is something that can be quite tricky when it comes to highly variable species. But our aim in doing the kind of species delimitation part of the taxonomy of Solanum is to get the whole of the genus at the same basic level of understanding the species. And then, of course, that then opens up a whole bunch of other questions about why are these things so variable in these different, is it habitat? Is it fixed genetic differences in different areas? You know, a lot of that level of work we haven't, I certainly haven't done, but it's there to do, and it would be fascinating because there are some very variable species.
Okay. What role has indigenous knowledge and local community played in your research?
Indigenous knowledge has been really, really important, I think, because in collecting in other countries, you always need to think about who's there before you, and what they know and what they would like to tell you and what they would not like to tell you. Now, I think indigenous knowledge is important, but local knowledge is also extremely important. Um, we we don't necessarily talk about ethnobotanical knowledge. Our work is much more taxonomic at the species level, thinking about what species are. But oftentimes people want to tell you about things about particular species of Solanum. But my work is not primarily ethnobotanically focused. But people are really important in taxonomy because it's people who you can talk to, who know where to find things. They have an intimate knowledge of the environment around them. Their understanding of nature is extraordinary. And it's wonderful to be able to share those kinds of things with people. So that's kind of, I would say that's the level of how Indigenous people have been important, I think, on the level of being people.
That's a great answer. Do you use BRM with vouchers? Does reference material stay with the vouchers?
BRM, meaning
Anne Harmon, can you clarify? Oh, this you may actually not be allowed to speak. So if you could throw it in the chat.
put it in the chat.
Um, yeah, we will come back to this.
BRM can mean all kinds of things.
Yeah. Wendy wants to know if would it be possible to get a copy of your collaboration slide? I think the one with the iceberg.
Oh, you know where there's a URL at the bottom. There's amazing resource which is done by these people, this group of people who are they're data scientists. So they're not taxonomists, they're data scientists. And it's called the Turing Way. And they're people who work at the Alan Turing Institute, which is here in London. And it's got all kinds of super interesting chapters about different kinds of collaboration. And some of them are about, I mean, one of the ones that struck me when I mean, I've always loved that picture, but there's another one which I used when we were thinking about our work on gender and naming is about forming groups of people and forming movements versus individuals. And it's just it's a fascinating resource. But definitely the URL, or just look for the Turing Way on Turing Way on the internet, and you'll find it. It's a great resource for all kinds of things.
So Anne followed up, botanical reference material for BRM. Do you use botanical reference material with vouchers? And does that
Well, I'm
reference
not sure what that
material?
means. I'm not sure what that means because a voucher is botanical reference material. I mean, a specimen. Do you mean a specimen? A specimen is a voucher. A specimen is a voucher. A tissue sample is a voucher. A silica gel collection is a voucher. So all of those vouchers are actually tied back. So this ties into what's known as the extended specimen. So if we have a specimen and we have lots of things attached to it, increasingly more and more information is attached to those voucher specimens, which means it's important to keep things in herbaria and in collections like fungaria, microbiology collections, all kinds of collections, because that's an actual object that someone can go back to say whether something is species A or species B, or whether it really does have the gene sequence that somebody published actually is the gene sequence. Because publishing a gene sequence is gone through a machine. There can be mistakes. It's easy to make mistakes in gene sequences. So
And
I'm not sure quite what that's getting at, but you know.
So we have from an anonymous attendee. When you speak of the phylogenetic relationship, the assumption seems to be that that is based on molecular sequence data, and morphological and life history traits are there just to be mapped onto the molecular tree. I know that is the current paradigm, but I think that is philosophically problematic. Should not a total evidence approach be taken and congruence of characters determine the tree, or am I completely off here?
No. I think some of these trees that we've done are based on phylogenomic data. They're based on phylogenomic data, but actually what's important is not so much mapping the characters on the tree, but also thinking about how those characters define monophyletic groups. So just mapping them on the tree is boring, but doing something where you can say, okay, which characters are labile across this molecular phylogeny? And then the other thing is that Hennig's principle of reciprocal illumination is super important. It's probably one of the most important principles in all of taxonomy, is that you look at something, and if it doesn't make sense with another data set, you go back and look at it again. So having separate data sets which tell you different things can be very, very powerful, because it allows you to kind of really do that thing of reciprocal illumination. Now, total evidence trees are incredibly useful. They're incredibly useful. They can be problematic because of the outweighing, the weighting of different characters that you have to create a total evidence tree. Creating even a phylogenomic tree for 742 or 800 species can be quite problematic. But I think we need to move away from thinking that putting morphology on trees is just about simple mapping characters onto trees, but it's about creating the possibility of looking at reciprocal illumination to look at where characters are homologous and not. So for example, if we had the fruit locule character, which is the fruits are multilocular, that happens in wild species of Solanums as well. And we mapped that onto the tree, and we saw that it was kind of in lots of different places. We might assume that that was homologous, that character was homologous. But the fact that it's not homologous, genetics told us that, not mapping characters onto trees. So it's really important to have multiple data sets and to think about things in a total way, as opposed to maybe total evidence. That's my opinion.
I want to thank Sandy so much. Sandy, I know you have another appointment to make it to as well. This has been wonderful. Wonderful to have you here, and wonderful what a great turnout this was. At our largest, we had 380, over 380 people here. It's great for our community. And
And I think the community is really important. I mean, it's
yeah,
important for us to stick together as a community and to think about how we can work better together to ensure that the science that we do, which is the bedrock of much of the rest of biology, actually continues. Maybe not in the way that it was done in the 1960s and 70s, but in a way that's fit for today's modern times.
100%. Thank you again. We hope to see
Thank
all
you,
of
Rick.
you again in a month. And thank you, Sandy. Take care,
Thank
everybody.
you.
Thank you, Catherine and Jennifer, for organizing as well.
Thank y'all.